everybody. It is really a privilege to be on stage with you folks today to talk about our sort of bespoke bio precision medicine theme. I wanted to start off by giving everybody a chance to describe how they sort of slot into the precision medicine world. So I think it's a world with many parts, and we all bring a sort of unique angle, whether it's academic or private sector or foundation or some combination of the above. So I'll kick us off by giving a little vignette about myself. And as some of you know, my life is sort of a precision medicine vignette, so it wasn't hard to choose one. But I'm myself at risk for genetic prion disease, a rapidly fatal neurodegenerative disease, and I learned this after having trained as a lawyer. My husband is trained as a transportation engineer, and we ended up leaving those careers in 2012 in order to retrain from the ground up in biomedicine to try to drive forward a therapy in our lifetimes. So it's been a whole thing, as you can imagine. But I guess one thing I would highlight about it that has been interesting to me, and prion disease is not ultra rare, it's not nano rare. It actually kills about 1 in every 5,000 to 6,000 people. Nevertheless, we are rare. And I think as we've navigated this landscape, we came in very naive. We imagined that our job was to discover the molecule that would effectively treat prion disease. And what has been impressive to me is the way that as we've progressed over these 10 years, the mission has just continued to widen. We were lucky to get really good advice early on that our job was way bigger than we thought it was. And that, especially in a rare disease, we didn't have the luxury of doing things in parallel. We needed to be looking at the idiosyncrasies of the biology of our disease and thinking, the luxury to do things in series. We had to do them in parallel. We had to be thinking about not just the molecule, but the patient population and how to get them and the natural history and the biomarkers and whether we had the right animal models and regulatory engagement and clinical strategy. And it was kind of going to be on us to assemble this platter. Mm-hmm. Maybe someday we would have a private sector partner, and in fact, we are lucky to be in that position now, but it was going to be on us to de-risk this platter of sort of different assets and different data in order to attract the partners that we would need to advance. So that's been our sort of experience of the special burdens of rare disease. Mm-hmm. But Gary, I'll throw it over to you.
Sure. Gary Hammer. I direct the endocrine oncology program at the University of Michigan, where we treat a variety of rare cancers of the endocrine system: thyroid cancer, adrenal cancer, and neuroendocrine tumors. Our foray into the precision medicine or rare space is predicated on the fact that these diseases are rare. Now, as I'll talk about in a session later today in a little more detail, as you all may know, the word rare is legislated as less than 200,000 cases per year, which means every cancer is rare except 3 or 4. So we really focused on trying to advocate for and get funding for and leverage our work to push forward therapies for what we would call the ultra-rare, the nano-rare, as you'll hear later from my colleagues, the ends of one diseases. And our foray into the precision medicine came as genomics became front and center, and we started learning more about the biology of the very rare adrenal cortical carcinoma, which is really where most of my energy is spent, where there's approximately 1 case per million, so perhaps 300 to 500 cases per year in the US, adult, maybe 30 to 60 cases in children per year. The only therapy we have is DDT, which is, as you know, a pesticide. Its derivative, DDD, is the only approved drug for adrenal cancer, while DDT single-handedly was banned from the earth in 1972, so that's a problem. So we have focused on trying to understand the genetics to lead us to new therapies. And to be brief, what we learned is that we had to work as a team. We had to really grow an international network to understand this disease, and that took about 10 years to get people to collaborate and engage together. And when the NCI started the TCJ project, as you'll hear about later today, they weren't initially looking at any rare cancers, But when they couldn't even find 200 colon cancers to sequence due to lack of collaboration, we convinced them that we could get 100 adrenal cancers overnight, and we did. And adrenal cancer is one of the first rare cancers that the TCGA took on. And what we learned in our laboratory, funded by philanthropy, by a grad student in the lab, that adrenal cancer came in 3 flavors, and one of them was driven by a well-known signaling pathway, which happens to be the acronym for this meeting, IGF, which also stands for insulin growth factor. And to make a long story short, that got me into this space because there were many drugs that targeted that pathway. They all failed in trials. Why? Because they weren't going after the right cancers. They were going after the main 5 cancers, when indeed the only cancer driven by IGF is adrenal cancer, with genetic forms, et cetera, of the disease. So we resurrected a drug from pharma, did a few phase 1, then a phase 2, then a phase 3 trial with IGF inhibitors. The drug always worked in a very small subset. We finally figured out, though, that it was only a small subset of patients that responded with a unique genomic signature, and slowly slogged getting that asset out of big pharma, got a nice $35 million Series A raise last year, and got this drug to be utilized in this rare subset of adrenal cancer for the first targeted therapy for that disease ever. So that's how we got started.
Awesome.
Okay, I'm Laura Sepp-Lorenzino. I'm the CSO of Intellia Therapeutics. That's a company in Cambridge, Massachusetts, where we're using CRISPR, genome editing, as the new frontier for developing therapeutics for multiple diseases, monogenic with the hope of going to polygenic or more common diseases for the very small populations all the way to the very large populations. My training, I'm a biochemist. I'm a drug developer, spent many years at Sloan Kettering and at Merck doing cancer biology. And I should have met you 25 years ago because I did work on IGF-1 and 2, and I was developing small molecule inhibitors in the wrong cancer, by the way. And since then, while at Merck, we transitioned into, at that time, what we were thinking was going to be precision medicine for oncology using nucleic acid therapies. And that led me to work on a mechanism that's called RNA interference, or RNAi, that can allow you to kill the messenger RNA that transmits the information from the DNA into proteins. That took me to Alnylam, which is a pioneer in developing RNAi medicines. Then I went to Vertex, where I had the privilege of collaborating with CRISPR Therapeutics on CTX001. And what we hope is going to be the first genome editing for ex vivo use for sickle cell disease. And then I got the call to join Intellia Therapeutics, and I'm glad I said yes, because it's my dream job. And what we're doing at Intellia is, as I was saying before, is really building all the tools, the platforms that allows us to scan the DNA, find the proper genomic location with high specificity, and being able to introduce different types of genetic changes as it's required by the disease. And in addition to that, for a drug, you need to have a means of delivering that CRISPR machinery in the body to the proper organ, to the proper cell type in that organ, and make it go to the nucleus where the DNA is. So, I have a large team working on that. And the last thing is that we're also using this technology ex vivo in cells that are either taken from a donor or from the patient, fixing the disease or fixing the gene or endowing that cell with new therapeutic orders and putting it back into the patients. So it's great to be here and hearing all the stories from patients, but also the technologies and the commitment of what are we going to be doing together to really bring genomic medicines as a reality to everybody. And we'll talk some more about that.
Thank you, Lyra.
Thank you. Well, hello, everyone. My name is Tracy Cole. I am the head of research at the N. Lorem Foundation. So we're an independent nonprofit focused on discovering, developing, and treating patients that have genetic mutations that occur in 1 to 30 people worldwide. And so we call these nanorare mutations. And Lorem is really focused and committed on using antisense technology with its versatility and selectivity to really help these patients that have nanorare mutations to provide experimental treatments. And we do this for free for life for our patients. And the reason we can do this is really based off of the past 30 plus years of experience in the antisense space, so focusing on the biology, the medicinal chemistry, and clinical treatment to really bring us to a time where we think that ASOs are well positioned to then treat these patient population. In addition, we've worked together with the FDA to establish several different guidances to really have a specific path for our nanorare patients for ASOs for the non-clinical, clinical, and manufacturing capabilities. And so excitingly, we've had since the past 2.5 years, we've had over 150 applications that we've received. We're working on 70 different active programs. And we just filed 4 INDs. We're very proud of that, and we're hoping to have 3 more by the end of this year. Yeah.
Fantastic.
Awesome.
Thanks.
Thanks, Tracy. It's a pleasure to be here. My name's Tim Yu. I'm a neurologist and geneticist at Boston Children's Hospital. My own personal journey to get me here actually began using genomics about 15 years ago to first try to discover causes of human disease, and we were lucky to be one of the very early adopters of high-throughput sequencing. Illumina was Alexa back then. There were a few more competitors back then, too, that have disappeared. And it was a heady time when we were using this technology to accelerate discovery. My laboratory then spent some time continuing that work and then extending it to accelerating diagnosis. At Boston Children's Hospital, we were committed to seeing the fruits of basic science go back to the clinic. And then in the last 5 years, we've had just the most incredible privilege to see how this type of genomic technology can be used to accelerate treatments. I'll tell a brief story. I apologize to the panel and others who may have heard it before, but it's really the inspiration for what our team focuses on now. I dare say maybe has also allowed me to connect with and provide supporting ammunition to folks like Tracy at Lorem and others who really see the value in this. But let me get to the story. In 2017, we met a young child who had just been diagnosed presumptively with a fatal neurodegenerative disorder called Batten disease. It was a bit of a mystery, however. They couldn't complete the diagnosis. They couldn't find all of the causative mutations. It's a recessive condition. They found one, but not the other. And our lab at that time, with an established expertise, not in therapy, but in diagnostics and whole genome sequencing, offered to enroll the patient and discovered that she had, via whole genome sequencing, a deep intronic mutation in the dark matter of the genome that is otherwise not interrogated by clinical sequencing. And as an illustration of the power of a finding and the diagnosis, that insight allowed us to understand what the diagnosis was, but also unexpectedly led us to a treatment opportunity. We began reading about how this mutation was working. We did experiments to confirm our hypotheses. It was altering the splicing of the gene. And we learned quickly from colleagues that there was this wonderful technology worked on by many scientists, including many at ISIS for 30 years, that was exceptionally well suited to addressing this type of mutation. And that lit a fire under us to try to develop a therapy for this mutation carried by this little girl. And in 6 months' time, our lab had prototyped such a medicine. It was working in the laboratory. We were naive enough to think that we could do it, and we were lucky enough to actually do it. And then I reached out to many folks in the field, oligo experts at Ionis, at Alnylam, at the Oligotherapeutic Society, and through really one of the most gratifying collaborative experiences I've had as a physician and a scientist, pulled together a team that took our prototype drug and saw it through necessary animal studies, manufacturing, and quality studies to get through FDA permission to start treating our patient 1 year from the time that we met her. And this effort has been discussed, and it remains a source of inspiration for us. It led to appreciable, tangible benefits for the patient, decreases in seizures, improvements in quality of life. Ultimately, sadly, it was too late to stave off the several years of disease that she had already accumulated before. But it's been a real inspiration for us, and is why I'm lucky to sit here with all of you today. From that case, the legacy of our first patient, Mila Makovich, her name I share with all of yours, with her family's permission, has led to the negotiations with the FDA that have contributed to the guidances that Tracy mentioned. It's opened a door for us to treat 5 other patients with individualized mutations with orphan diseases that we've been able to design with copious help from many. And I hope is the beginning of us really grappling with what we are going to do in this new age when the technologies for intervening with high ceilings actually are targeted at the mutation level. They change the focus of the conversation not to the disease and to large population policy questions, but to individuals in a way that as a physician scientist I actually find very satisfying, very gratifying, and very empowering.
Yeah. Fantastic. There is so much experience on this panel, and I wish I could pick your brains all day. But I want to pick up on some of the themes that Tim was just mentioning. There is this sort of policy and regulatory dimension to what we're all trying to do. And I was, speaking earlier about the constant expansion of my mission. I think one thing that is striking to me at this phase and was underappreciated by me 10 years ago is that we have the scientific challenge, and I don't want to downplay it, and I certainly don't want to speak from a place of hubris, but I think we are really lucky in my disease, in prion disease, that we have a very clear genetic blueprint and a very strong therapeutic hypothesis. I now, at this stage, look at how close we are scientifically and realize that there is a whole new failure mode, this sort of like valley stretching out in front of us where we may languish for a long time, where we are here scientifically, but are ensnared in the sort of human-made systems and logistics. And it is terrifying to me that that is the way we may run out of time. I never thought about this 10 years ago when we launched this quest. It was not on my mind, but now I think about it every day. So that is thing one. The second thing that I was just reflecting on is that I love the work that you guys, Tracy, Tim, that folks are driving forward in terms of N of 1 best practices and the examples, the heroic examples that have arisen that we can look to for inspiration. I think as we broaden the application of N of 1 thinking, it's going to have to encompass the idiosyncrasies of so many different diseases and how their biology sort of has to steer the process. And for prion disease, we're up against this particular obstacle that it is exceptionally rapid. So the average patient will die within a few months of their very first symptom. This has led my husband and I to focus deeply on prevention. That's its whole other interesting, struggle and path-making journey. But what I'll say is, when I look at N of 1 examples that have come to light to date, Tim, you mentioned the idea of being too late. In prion disease, in order to motivate N of 1 urgency, would we have to be looking at a very sick patient? And if we are looking at a very sick patient, isn't it inherently too late? What do we do about diseases that are like a lightning strike? Because I feel the urgency right now. And I would happily go in front of somebody and say, this situation, this near certainty of a fatal neurodegenerative disease striking me down in midlife, which I am in, by the way, that could strike at any moment, certainly justifies N of 1 calculus. But I think people would look at me, you guys tell me, I think they'd look at me and say, you're not sick. And how do we get past that? So I want to throw it to Tim first and then work our way back down the line.
Sonia, you've put your finger on a really critical question that has no single answer. And I know that you and Eric have thought very deeply about this, and the answer that you've come to is absolutely appropriate. And it's that level of personalized thinking which is required to adjudicate these situations. I'll offer an answer by way of anecdote. The second patient that followed after our Mielison work was actually a young baby girl who was born with that abnormal finding on newborn screening that actually initially raised a concern for immunodeficiency, a severe combined immunodeficiency. Turned out that was not the case. It turned out she actually had a neurodegenerative condition that, for technical reasons, sometimes causes false positives on newborn screening. And it was a fatal condition for which she had minimal symptoms, but was predicted to die by young adulthood. It was in the pre-symptomatic phase that you're describing, Sonia. And we sat and wrestled and discussed this with the patient's, the family foundation that supports these families, with our clinical colleagues, experts in the disease, ethicists, and came to terms with what we ought to try to do, and realized that there is a window of opportunity to intervene early enough to impact the course of disease, which is completely separable from manifestation of symptoms. And I'll say for an N of 1, well, this was followed by an N of 2, another second patient with the same condition. The FDA, working in concert with us, agreed with our calculus and gave permission for that intervention, that experimental intervention, to go forward, and we are now 3 years into it. So I think that there is room to accommodate the type of struggle and decision-making that you're describing, Sonia, and we shouldn't assume that it has to be always the same way.
I love that.
Okay, down the line. Okay. So at Lorem, we're also realizing that there's so many more nano-rare patients than we really originally ever thought would even be possible. And so we're focusing on trying to help the patients that we can today. There are patients that come to us, they've had more, maybe 7, 8 year diagnostic journey. They feel helpless and hopeless. And so we do our best if ASO strategy is amenable for our patients to help them. But we absolutely realize that there are certain patients now that we cannot help. And so we also try to learn as much as we possibly can from the process, from all of our patients coming through, so that we can do better for our next patients that come in, increasing capacity, improving efficiency, trying to, especially working with Illumina, it's amazing, right? Making genetic screening more accessible to everyone is really gonna help us identify patients earlier, so they can come to us before they have progressed so far into disease. So yeah, we're just trying to take and do our best that we can now, and then improve for the future.
You know, and I think that what you guys are doing is amazing. And, you know, what's the beauty of it is that you're leveraging 30 years of research and human experience on the antisense technology, you know, and as drugs, right, for multiple different modalities, different organs, different routes of administrations, dosing. So, you know, when you're thinking about regulatory pathways, right, there is this enormous corpus of data that supports going now to the N of 1. For new technologies like the ones, you know, I'm doing for genome editing, we still need to build that. So, you know, although, you know, from the science point of view, you tell me what the mutation is, and I can tell you in, you know, a few minutes, literally, what is a change that we need to elicit to fix it. What still is missing is that, preclinical data, the analytical, the manufacturing, the toxicology, and the interactions, so our regulatory agencies, whose job is to make sure that there are standards and that, patients are safe, right? That it will be that level of confidence that we're ready to bring that to the N of 1. And the goal is, for me and the other companies in the CRISPR space, right? I think we're all doing an amazing job at coming up with new ideas and, new utilities, new applications of the technology. It's just doing responsible, good science and clinical science to build this corpus of data that will pave the way to N of 1 therapies.
Laura, one thing that you just said that I totally see in the world and just find so interesting is that there is a degree to which N of 1 kind of has to come second. Mm-hmm. And that's so interesting because it is actually the paradigmatic case where the risk-benefit would ask you to charge ahead, right? And yet I totally agree with you that ASOs have been able to explore this space because we stand on such a corpus of data gathered from larger patient populations. So I'm not sure what to do with that, but I think it's an interesting paradox.
And the other one is AAV gene therapy, right? So the NIH, FDA, and a number of organizations have the Bespoke, gene therapy consortium. But there, again, they're building, right, on this enormous amount of data that exists. I think that as a community, we need to do this faster, right?
Yes.
In a collaborative manner, so we can bring these therapies, to patients much, much sooner than 15 years from now.
Oh, yeah. Here's hoping. Absolutely.
So let me close out this question by switching from a little bit of science discussion to policy. And I charge everyone in this room, including everyone on the stage, to engage and be bold and get out there to help change policy, because that's the only way we're going to advance and incentivize work in the rare disease space, rare cancer space, let alone the N of 1 space. So my vignette will be that I spent about 2 years on Capitol Hill working with Ted Kennedy when he had his glioblastoma, and his chief of staff, who actually had adrenal cortical carcinoma that I study and treat. And we spent So he worked with Bailey Hutchison. It was a bilateral committee on the HELP committee, and we spent 2 years drafting legislation for rare cancer support, to advocate, to support, to get patients to the right doctors, to incentivize companies. It's a different kind of hard work, but it's all of our responsibilities to make that happen. And healthcare reform subsumed that bill, even though we were on Capitol Hill watching the bill announced in Congress. Healthcare reform took over. Slowly but surely, however, the word about rare diseases started to take hold. What you should know is that the word rare is legislated as less than 200,000 cases per year for any disease. That is a challenge for truly rare diseases, be they Ns of 1 or nano-rare or ultra-rare. When I tried to advocate for those types of words, we got big pushback from certain communities because we were taking away from that broader rare population. And slowly but surely, however, and I'll discuss this in more detail later today, slowly but surely, despite big challenges, genomics took over. Genomics defined all of us as rare. Every individual is an N of 1, and we're learning that, and we're getting there. But it takes a village to make that happen at the level of policy, because we have to incentivize and leverage our work for both academic funding and for funding of industry. So I charge all of us that as our homework assignment for the next 5 years, to do our best to be bold and fearless when you can in the regulatory and policy space.
I want to shift us into another tricky area that we are certainly not going to solve in our remaining moments here today, which is rare versus common as a business problem. So here I am championing a rare disease that's not, not so super rare, 1 in 5,000, 1 in 6,000. And it puts us in this interesting gray area where some companies see us as commercially viable, some don't. So we engage in lengthy courtships and we assemble platters and we hope and pray. For those of you representing the public sector, Lara, Gary, what would it take from your public sector or private sector perspective to take on a disease like mine? Because, I mean, we talk about gene editing, like we talk about vectors that maybe someday hopefully can reach the whole brain. These things are my dream. And my nightmare is that it exists here on Earth, but I can't bring it home to my community. And to Tim and Tracy, I mean, you guys are in this space of serving the very, very rare disease need, but you can't possibly keep up with everybody who's knocking on your doors. So how do we scale? Anybody jump in?
Maybe
go.
Please, Lola. Okay.
Too polite.
I'll take first. I'll take first step then. I think that one of the bold and perhaps idealistic answers, I think, that I hope will actually come true, is that I think that we have an opportunity to begin. We have an opportunity to work with tools that have the potential to be robust enough to get us out of the one drug, one disease mindset. And I look ahead to the time when we can define a process which is robust enough, efficient enough, and safe enough to develop interventions for patients that aren't necessarily, we won't be naming every molecule that we deliver to a patient. If it takes 200 drugs to treat 200 patients, we'll run out of names. But what we will do is define solid processes that allow us to make them in a reliable and predictable manner, and that process will become the focus of attention. An over-easy analogy that I sometimes draw on is the fact that we have personalized care for patients that we deliver in hospitals every day. Surgery, transplant. These are medically complicated interventions with severe risks, very careful risk-benefit analyses that have to be tailored to the individual. The tools, of course, are predictable, scalpels or lasers or whatnot. But if we can aspire to reach that level of expertise with the tools that we've got, knowing that we aren't there yet, I think that approach might allow us to break down sort of the traditional ways of commercializing individual molecules.
That is a lovely vision.
From my perspective, at the company, so again, for what we do, which is genome editing, at Intellia, we were the very first who did genome editing as a therapeutic in vivo, meaning that we infuse the CRISPR system into the vein of, multiple people now, over 30 people with 2 different diseases, and really showed very, very clearly that CRISPR actually works as intended, that, the pharmacokinetics, the pharmacodynamics, the effect is as expected, as well as the duration. CRISPR editing, if you fix a gene, that gene, once it's fixed. There is not going back. So, the promise of one and done cures. And that's a huge responsibility because being first, we need to ensure that, as Tim was saying, right, that, this works as intended with the, safety, the reproducibility, the, performance that you want to have it. So that's our focus, right now. We're working on monogenic diseases, so they are not ultra rare. There is, one case, 25,000 to 50,000. In another one is up to 500,000 people. So that's, big numbers. But, my hope is that that's going to show us that we can actually do this safely. And once that we have that data, it will allow us, whether it's us by ourselves or in in partnerships with others, right, to bring these, molecular surgery to everybody. And this is also building the tools that allows you to do that, right? Because, right now we can, use, in our case, a non-viral particle that takes CRISPR to the liver. So if there is a disease that's driven by a gene that's either expressed or missing in the liver, we can fix that. Absolutely, we have shown that. But there are many other diseases that require changes in other organs, including prion disease.
Yes.
And there is how do you deliver it to every single cell, right? Or to the majority of the cells to elicit the pharmacological change you want to do that.
I just hope you'll call me when you know the answer. I know we're not there today.
We're not there today, but there are many people working together, and it's really trying to find those synergies, right? That can allows us to mix and match.
Gary's getting antsy over here.
Gary,
Tell us, Gary.
come on.
I'm not getting antsy. I'm looking at the
Yes.
rare versus common. So you've heard a bit about platform development and exploiting a platform across multiple diseases, be they rare or common. The other way, of course, which is on your mind, is to exploit a biological pathway and to understand the biology of a disease and to realize that the biology may be critical for a rare disease, but may be exploitable in a common disease as well. It's very difficult, as I have found, to oftentimes to find investors on the side of industry for a ultra-rare disease, however we want to define it. However, what I always get asked is, yes, but how will this help this common disease of hypertension or something like that? And so if you can exploit the rare for the common, you don't want to shy away from that, even though we all are passionate about sometimes our disease or our platform. You must not be afraid of that, and indeed, that is how we ultimately got funded, because the very pathway that we study in our rare disease that is critical for adrenal cancer actually drives a recently discovered common disease. And so we exploited that to get funding for common to do rare. And I don't want to leave that off the table in terms of a business strategy, which
Absolutely.
is part of the discussion here.
Gary, I embrace it, and I so celebrate the cases where it works. And I also, my heart pushes back against it in some ways. So I think both are true situation, but I fear a world in which we have left rare diseases to a lottery where some have a nexus, a genuine biological nexus with a common disease, and some don't. So I don't have the solution to that.
I don't argue with you. It's a really heartfelt problem for us all.
For sure. Tracy, jump in.
So at Lorem, we feel that we can help our nano-rare patients in a nonprofit model. That's what we're dedicated to, and to provide our therapies for free for life. But this is dependent on the fact that we have this amazing network of corporate partners and private donors and companies that work with us to help us purchase equipment, and just like Illumina, helping us provide whole genome sequencing, which is absolutely critical for every one of our patients to develop the individualized therapies. So in a nano-rare sense, we're working to do that. However, at the same time, like we're saying, so we have these nano-rare guidances from the FDA, but what about the guidances from the 20 to 200 range, right? And so now we're trying to think forward to that too, for the future, to make that a reality, and figure out what does that look like for all of our different guidances. But yeah, we think it can be done in a nonprofit model.
Absolutely.
Thank you, guys. As I said, so much wisdom to share here. I do now want to move us into my patented lightning round. And
It didn't work out yesterday.
well, here's what I'm going to say. I've intentionally left us very little time. So you're going to have to actually be lightning, unlike some people we know. Okay. So I want to cue us up by just acknowledging that this is hard work. And I know, and I think you guys do too, that in my heart, I know I'm pointed in the right direction and that this work matters. And there's a grounding in that. But I sometimes find myself wondering, in the fray, in the weeds of the day-to-day, I find myself thinking, if this is the right thing, why doesn't it feel better?
Yeah.
And it doesn't feel better because it's hard. So in that frame where we've all chosen to stake our flags and fight our battles, what keeps you hopeful? Gary, kick us off.
Sure. The arc of human history bends towards justice, to quote
Love it.
President Obama.
Love it.
Science keeps me hopeful. Rational thought and science keeps me hopeful, and patients keep me hopeful. We are all patients. We will all not escape this earth without being patients, and that gives me extreme hope as patient engagement takes center stage in all that we do.
I cannot beat that.
Yeah. Yeah, I agreed. I agree. Our patients are what brings us here, what keeps us passionate, what keeps us motivated to do better, to always improve our processes, improving capacity, efficiency, everything. The reason we're all here is because of our patients.
I think I'm going to continue the theme and echo that and amplify it. I think that our work in the last 5 years has been some of the most rewarding professional experiences that I've ever had. Engaging deeply with families, working with amazing collaborators who are so incredibly generous because they see we're at a point now where the science is at the bedside, and everyone welcomes this opportunity to contribute something that has tangible good to someone that they can see. I think that that power, the ability to democratize doctors arguably get to see this maybe every day in clinic interactions, but to be able to democratize access to that experience to everyone throughout the community and to see that it's actually contributing to good for patients, it's an incredibly enabling and powerful tool to incentivize all of us to do more.
I love this. Tim, in our prep, you had a nice analogy about a North Star that can sort of guide us through this territory. Tell us about that.
As an academic, we know that life is not always just idealistic pursuit of truth and knowledge. There's sometimes sharp elbows and sometimes competition, and that actually competition serves great good. In our policy decisions to bring drugs to the market, we also know that it is not always simple, clean, clear. Business questions sometimes necessarily intrude. As the world of science and pharmacology and drug development collides with parents, advocates, and patients, and we're doing good, that North Star is blazing bright right in front of us. And you cannot look away from it. And I think it's just going to drive more and more good behavior.
Is that one more thing?
Gary, jump in.
I love that analogy. And I just want to riff off that and say, if we let mission follow, if we let mission lead, the rest will follow.
Yeah.
Exactly right.
Okay. We really can't end on better words, so we're ending right there. Guys, if you let mission lead, the rest will follow. Thank you, everybody.
Thank you.
Thank you.
Thank you.